Hello everybody, thanks for joining today's webinar hosted by Bio-Rad Laboratories. Today we'll be discussing microbial safety and the testing of cannabis. My name is Kevin Moore and I am the Technical Sales Specialist for the Food Science Division of Bio-Rad Laboratories. I'll be the facilitator for today's call. Our presenters today will be Ms. Emily Savage and Sharice McDoula from CW Analytical Labs in Oakland, California. The agenda for today's webinar will cover the following topics. Emily and Sharice will discuss the current scenario and challenges in cannabis testing, Cannabis Microbiology 101, how microbial testing is performed, the importance of validated methods, and learnings from a cannabis testing lab. Kevin will review the following information: a company overview of Bio-Rad Laboratories, IQ Check, a real-time PCR technology, Bio-Rad's cannabis product portfolio and features, IQ Check protocol, chromogenic auger as a confirmatory tool. We'll also discuss our next webinar, and we'll finish things off with a question and answer session. And now I'd like to hand things over to Sharice and Emily.
Hello, I'm Emily Savage. And I'm Sharice McDoula from CW Analytical Laboratories, and welcome to our webinar.
All right, I'll start with an introduction to CW Analytical and the cannabis testing industry in California. CW Analytical is one of the oldest and most trusted labs in the state. We're located in Oakland, and we service the entire state of California. We were co-founded in 2009 by a chemist and a microbiologist to aid cultivators in producing clean, safe, and consistent cannabis and cannabis products. Today, we offer a full suite of analytical testing services, including everything required by the Bureau of Cannabis Control. In addition, we offer and encourage research and development collaborations with manufacturers and producers of any product who desire to know more about their product and contribute to scientific inquiry, because the labs truly can't do it alone. Early on, testing centered heavily around potency to determine price per milligram of THC or CBD. This trend began in dispensaries, and soon after, individual cities and counties began passing their own testing regulations with focuses on other quality aspects like pesticide, microbiological contaminants, and residual solvents. In 2018, quality assurance testing became mandated by the state of California. Throughout that time, CW Analytical has remained community and education focused. Since 2009, we have helped many producers troubleshoot quality issues with their cannabis or cannabis product, whether that's dosing and infusion, scaling, loss factors and manufacturing issues, mitigating and locating microbial, heavy metals, or pesticide contaminants, holding educational seminars. We're happy to explain data, give it another look, engage in collaborative and scientific conversation with our community, our clients, and each other. So with that said, let's dive in. All right, let's start with a brief overview of cannabis history, touching on some major milestones. Cannabis has been attributed to a variety of medical uses since the oldest Chinese pharmacopoeia, and there have also been historical accounts of medical use in ancient Egypt, India, Greece, and the medieval Islamic world. Medicinal preparations of cannabis became available in American pharmacies after Dr. William Brooke O'Shaughnessy introduced its use to the Western Hemisphere in the mid-19th century, and it was added to the U.S. pharmacopoeia. By the 1900s, cannabis quote-unquote drugs were being manufactured for use as antispasmodics, sedatives, and analgesics. In 1906, the Pure Food and Drug Act was passed by Congress as the first of a series of consumer protection laws, which led to the formation of the Food and Drug Administration. The Act implemented a series of labeling requirements and was the first venture towards quality assurance of medicines, including cannabis. However, by 1913, the California Board of Pharmacy had outlawed cannabis in the state via an amendment to the Poison Act, and the following Marijuana Tax Act of 1937 banned it across the nation. Fast forward to 1996, California passes the historic Proposition 215, or Compassionate Use Act, which indicates a reemergence of acceptance of cannabis for medicinal purposes in the state. This was the first state to re-legalize medical cannabis, and it was passed for a huge variety of illnesses including cancer, anorexia, AIDS, chronic pain, spasticity, glaucoma, arthritis, migraine, or any other illness for which marijuana provides relief. The legislation did not dictate any set of rules regarding the safety or efficacy of any of the medical cannabis products dispensed, and it allowed for a diverse array of brands and products to develop, many of them beginning in home gardens and kitchens. Today we know that under the Controlled Substances Act of the DEA, cannabis is classified as a Schedule I drug with no currently accepted medical use or treatment. Despite the federal government's stance on cannabis, 33 states and Washington, D.C. have passed legislation to support the medical use of it, and 10 states and Washington, D.C. have legalized cannabis for recreational use. A 2018 Gallup poll shows that 66% of Americans support legalization. The figure on the right shows all of the states that have adopted either medical or recreational legislation to support the use of cannabis. All right, let's discuss some challenges faced by the cannabis industry, of which there are many, both nationally as well as locally here in California. Nationally, this can be summed up into a few major points, the first being a lack of oversight. Other agricultural industries are regulated by federal agencies like the FDA and the USDA. This leads to a lack of support for scientific research. I believe there's one university with federal access to cannabis, and the cannabis that's grown there is not representative of what we see in the marketplace today. Each of these compounds into each state developing their own unique set of rules and regulations. I'll touch on what this means for quality assurance and laboratories specifically in the next slide. Locally here in California, we have some huge barriers to entry, including taxes, permitting and licensing fees, zoning laws, local bans, as well as access to banking. California is also in a unique position to try and bring a mature, well-developed industry with a huge economic scale into a highly regulated marketplace. One of the final challenges faced by the California industry is maintaining the community-minded aspect of the industry as it continues to grow. This was an industry that was started on a grassroots movement, and since legislation in 2018, we've seen a huge loss of some foundational community members, and it has certainly presented itself to be a challenge for this industry. So what do all of these challenges mean for quality assurance of cannabis and the laboratories that provide this information? We face our own set of unique challenges, the first being a lack of standard methods. There are no published standard methods for cannabis, which leaves each lab to its own devices to create their own unique testing methods, which might be okay if cannabis were one matrix. It's often referred to as a singular matrix, but is truly an umbrella term, which includes but is not limited to plant material like flour and shake, concentrated material such as hash, keef, wax, butter, shatter, live resin, rosin, sugar, and the list goes on. And even an even more expansive list is that of infused products such as foods, brownies, cookies, other baked goods, candies, lozenges, mints, tablets, gummies, capsules, teas, sodas, topicals like salves, creams, balms, lotions, patches, bath bombs, suppositories. The list is constantly expanding, and it often leads to a lack of robust R&D testing prior to compliance testing. It's critical to understand a product and a matrix, of which there are so many unique ones, prior to entering a compliance testing situation, not only for the manufacturer and producer, but for the laboratory to provide the most accurate data possible, which in turn gives the consumers the most accurate data possible. In addition, cannabis faces some pretty rigorous testing standards. At this point, it's more regulated than most food at the grocery store and over-the-counter medications in regards to pesticide, heavy metals, and residual solvent allowances. In regards to potency labeling requirements, the standards are more stringent than many pharmaceuticals. In addition to this, some testing standards are bleeding-edge technology that are not employed in other industries and not in routine settings. This leads to expensive testing costs that labs must pass on to their clients. All the while, the demand for a laboratory's data drives the industry and has driven some laboratories' turnaround time to unrealistic timelines. These are just a few reasons to consider a lab with a minimum or a slightly longer turnaround time to protect all stakeholders to the data. This figure shows a summary of the required testing for inhalable and non-inhalable cannabis products in California. As you can see, the list is quite comprehensive. The pesticide list contains 66 analytes alone. Today we'll be focusing on the microbial impurity section, including Shigatoxin E. coli, Salmonella, and 4 species of Aspergillus.
Indication of cleanliness of a product or process. We can use tools such as APC and yeast and mold Petri film to determine how much filth a product contains. Without knowing what exact organisms are present on the product, we can use a general survey count on all aerobic bacteria and yeast and mold organisms present. Liability protection. Having general knowledge about how filthy your product may be can tell the manufacturer, producer, or cultivator whether their process or environment isn't suitable and can prompt for changes that need to be made. This can be used for liability insurance purposes. Detection and mitigation of microbial contamination. Once contamination is detected on your product, you are able to learn more about your options and about proper handling of your product and take steps towards remediation to try and avoid the issue in the future. Per regulations of the state of California, the testing for certain microbiological contaminants is required. This requirement includes STEC, Salmonella, and 4 species of Aspergillus. Most types of E. coli are harmless, but some groups of E. coli, such as Shigatoxin-producing E. coli, also known as STEC, produce toxins that can cause illness and may even damage your digestive tract. State regulations also include testing for Salmonella. Salmonella is easily outcompeted by other organisms and requires a special cultivation process. This is why longer turnaround times are necessary for the robust testing methods and procedures. Taking extra time to test properly also ensures that the results produced are correct and accurate data. Lastly, state regulations require speciation of Aspergillus. The 4 species of Aspergillus required to test for are Niger, Pterius, Flavus, and Fumigatus. It's important to note that no other quality assurance setting requires the speciation of Aspergillus. Therefore, there are no methods developed in a quality setting to test for this. This makes it difficult to create a standardized testing procedure for the industry. Since the industry is still fairly new with little research performed, we do not yet know the exact impacts of pathogens on cannabis. So we rely on the information we know about the impact of pathogens on contaminated foods on human health to help create the regulation. APC is a Petri film that provides a total aerobic bacterial plate count indicated by red colonies. Yeast and mold is a Petri film that provides counts of yeast indicated by small blue-green colonies and also provides counts of mold indicated by large, variably colored colonies. E. coli plates provide both an E. coli and coliform count. These bacteria are indicated by red colonies with gas bubbles present for coliform growth and blue colonies with gas bubbles present for E. coli growth. A color change to yellow or orange also indicates the presence of Pseudomonas. Petri film is a great way to determine how clean or dirty a product may be. It also provides a cost-effective, convenient, and reliable method for testing in rapid time. Manufacturers and producers can benefit greatly from these indicators, using them as a cheap insurance policy. With California regulations, there are only 6 organisms required to test for and only 2 for non-inhalable products. If there's some bacteria present on a product that's not on the list of required organisms to screen for, that leaves vulnerability for the manufacturers and producers. APC and yeast and mold can aid in preventing that from occurring.
So you might be curious about what foodborne pathogens need to survive and grow. This is summarized with a mnemonic device used in the food service industry called FAT-TOM. This stands for food, acidity, temperature, time, oxygen, and moisture. These are the basic 6 conditions that foster the growth of foodborne pathogens. We can go through each of them individually. The first is food. This ensures that there are sufficient nutrients available that promote the growth of microorganisms. Protein-rich foods like meat, milk, eggs, and fish are the most susceptible. The next condition is acidity. Foodborne pathogens require a slightly acidic level of pH and thrive in basic conditions. Temperature. Foodborne pathogens grow best in temperatures between 41 and 135 Fahrenheit or 5 to 57 degrees Celsius. This range of temperature is referred to as the danger zone. They thrive in temperatures that are between 70 and 104 degrees Fahrenheit or 21 to 40 Celsius. The next condition is time. All food, whenever it's prepared, should be removed from this danger zone within 2 to 4 hours by cooling or heating. Oxygen. Almost all foodborne pathogens are aerobic and require oxygen to grow. There are a few anaerobic organisms to consider as well. The last condition is moisture. Water is essential for the growth of foodborne pathogens. Water activity is a measurement of the water available for use by microbes and is measured on a scale of 0 to 1. Foodborne pathogens grow best in foods that have a water activity between 0.95 and 1. The BCC regulations require that all semi-solid and solid edibles have a water activity below 0.85, and all flours and pre-rolls have a water activity below 0.65. All right, so to bring it back to cannabis, let's talk about the process from cultivation to use and some potential microbiological risks along the way. During the cultivation or growth process, indoor and outdoor plants are exposed to a variety of bacteria and fungi. Many people are using probiotics and pest controls, and this wouldn't necessarily introduce bad bacteria, but of course it's always a possibility. Additionally, the use of compost teas can be risky, especially if applied foliarly during the flowering cycle, and properly prepared compost could introduce pathogens. In addition, inappropriate growing conditions related to temperature and high humidity can support the growth of pathogens, both plant and human. However, the biggest risk that we see for microbiological contamination tends to be during processing of cannabis, so drying, trimming, curing, and further processing. A lot of this is related to GMP or good manufacturing processes, which all really comes down to cleanliness. Post-harvest flower can pass through many hands on its journey. If introduction of pathogens does occur, they can survive desiccation all the way to the consumer. Some tips that we give to prevent contamination include no carpet in your facility, having rooms for entering and exiting the building or the facility, not wearing street clothes into grow operations or processing rooms, use of non-porous surfaces or stainless steel work surfaces that are frequently cleaned with 70% isopropyl alcohol, not allowing any pets in the facility, humidity and temperature controls, frequently checking your HVAC system to clean and change filters on a schedule, discarding loose trim or plant material with any visible rot or fungal growth, frequent cleaning of trimming scissors, employing the use of beard nets and hair nets for your staff that are touching the cannabis, trimming sequestered batches into clean bins, and good glove changing protocols. If it touches your hair, your skin, your cell phone, or clothes, then they need to be changed. If a consumer were to use cannabis with a pathogenic microbial contaminant, it could be ingested in a few different ways, one of them being inhalation. So smoking and vape temperatures generally only kill non-spore-forming bacteria. This has not been scientifically investigated and would be a really good study to perform. Additionally, digestive and eating of edibles is often a way that people consume cannabis and could be introduced to foodborne pathogens. One way to check this or prevent this is employing the use of aerobic plate counts and total yeast and mold counts on edible products. Additionally, cannabis is often applied to the skin in the form of topicals, and with all of them on the market, this is another potential area for quality assurance risks. Let's discuss some of the previous reports and literature of microbiological studies performed on cannabis so far. There have been a few anecdotal reports linking both salmonella and aspergillosis to cannabis. Most of these studies were performed in the medical field, seeking epidemiologic causation to a patient's illness. This can be an extremely difficult task to perform whenever we're considering everything that a patient is exposed to on a day-to-day basis, including food, cosmetics, air, and water quality. Aspergillus spores are ubiquitous in the environment, and salmonella is generally a foodborne pathogen. In all of these studies, there were no pathogens detected on cannabis in question prior to consumption. In addition to this, any pathogens that were isolated from the cannabis after the fact are not representative of the quality of the kind of product that we would see today. The cannabis that had salmonella on it was brickweed that was cut with manure, so it's really not representative of something that we would see on the shelf today. In addition, many of these studies were published at the height of the drug war. Beyond this, we have a few fungal surveys that were performed by McPartland. These were sort of scratching the surface and starting to dive into the potential natural microbiome of cannabis, potential plant pathogens, and there's a lot more research there to be done. So essentially, there are very few or no empirical, peer-reviewed, published research on microbiological aspects of cannabis, which is something that I would expect to change later on this year.
Microbiological testing is performed in a variety of ways. None of these methods are great as standalone tools, but they all complement each other well, allowing you to make scientific decisions based on multiple sources of data. Feature film plate count, including APC and yeast and mold, give us a general survey of cleanliness, counting colony-forming units. Specific organism screening yields on PCR give us a negative or presumptive positive result. Selective enrichment broths and media. For STEC, we use MTSB for enrichment and Shabam agar for the medium. And for Salmonella, we use BPW for pre-enrichment, RD broth for further enrichment, and XLD agar and TSI slant for the media. We use Bio-Rad's IQ Check STEC kit and Salmonella kit for our biochemical analysis and utilize MALDI-TOF as a final confirmation step. All data shown on this graph was collected using 3M yeast and mold Petri film. Yellow represents hash, light blue represents keef, green represents shake, dark blue represents pre-roll, and brown represents flour. Hash and keef have little to no fungal load. We very rarely detect fungal organisms on these matrices. We also find that less than 1% of solvent-based concentrates show growth of any fungal organisms. Matrices that we see over 10,000 CFUs per gram are usually on pre-rolls and shakes. Fungal loads on most products are generally less than 5,000 CFUs per gram. All data shown on this graph was collected using 3M APC Petri film. In this graph, light blue represents flour, orange represents hash, gray represents keef, yellow represents shake, and dark blue represents pre-roll. We found that the aerobic bacterial load on most products are less than 50,000 CFUs per gram. We see lower percentages of samples showing counts of colony-forming units between 50,000 and 500,000 and at a pretty consistent rate. We also see that hash yielded the highest results for colony count on APC. Hash is mechanically processed and can concentrate any bacteria present. There's no opportunity to kill bacteria because it is processed in cold water. Shake is also high in CFU count. Material made with shake typically is not treated well, including pre-rolls. But we could set a really high limit for APC still, ensuring great passing rates. It's a great way to get a general survey of cleanliness of your product, warranting potential follow-up testing due to high-yielding results. Most products will pass. It's a cheap route in terms of testing material needed, and it's an extra protection with an insurance policy for cultivators and producers. This data was gathered from cannabis samples consisting mostly of flowers. All samples were processed through selective enrichment growth of gram-negative bacteria. We were investigating for Salmonella in particular. The samples were inoculated in pre-enrichment broth, run on PCR, inoculated onto XLD agar and TSI slants, and run on MALDI-TOF. This pie chart shows the diversity of the most common gram-negative bacteria we found on cannabis. In blue, we have Enterobacter with 53%. In green, we have Klebsiella with 26%. In orange, we have either Shigella, Escherichia, Citrobacter, Chronobacter, or Pantoa with 26%. And in purple, we have Pseudomonas with 17%. We tested for all genera of Salmonella and not species-specific because we were most comfortable with that level of identification. We were studying the occurrence of Salmonella, and no Salmonella was ever detected throughout our study. CW Analytical uses Bio-Rad's IQ Check RT-PCR to screen for Shiga toxin E. coli and Salmonella. The PCR is used as a screening tool post-pre-enrichment. Any presumptive positives are sent to culture-based follow-up. When evaluating technologies, we chose Bio-Rad for a few reasons. First, for their robust validation protocols, utilizing a wide range of live culture organisms. It's very important when validating to use live culture. Performing an evaluation based on nucleic acid spikes doesn't test your DNA extraction process, which is crucial to the success of your assay. Secondly, the use of pre-enrichment protocols. Pre-enrichment is time well spent when looking for pathogens. This gives you the utmost confidence that your product is free of these pathogens, especially Salmonella, which is often outcompeted by other organisms. Next, the kits come ready to use, and it's really easy to train a technician. This saves a great amount of time on training and removes the stress of having a limited amount of employees who are able to perform the procedure. Lastly, their amazing customer service is truly appreciated. They make themselves completely available to assist if any issues may arise and are always willing to do meaningful collaborations to help make great science resources accessible to all.
All right, let's end our section with a brief recap of what Sharice and I discussed today. To start, the microbiological risks on cannabis and cannabis products are still being determined and should be determined by product type, manufacturing involved, mode of ingestion, and naturally occurring microbiota and pathogens that are detected or isolated. Generally, there is very little bacterial or fungal contamination on solvent-based concentrates, and the majority of cannabis and cannabis products do not contain high microbial loads. For these reasons, quality indicator surveys such as aerobic plate counts, total yeast and mold counts, and E. coli coliform pseudomonas surveys can be used by manufacturers, producers, cultivators, and distributors as an affordable and rapid way to protect their liability and help keep consumers safe. Whenever considering all analytical data, your method is critical to generating accurate and precise data that you can be confident in. Specifically with microbiological methods, the use of pre-enrichment times and confirmatory data are extremely important aspects of this. And this is not to say that it should take longer than needed to release results, but it is to say that if it takes 1 or 2 days longer, it's time well spent to support not only our science, but by extension our clients and the patients or consumers of the cannabis that we test. In this same tone, validation of microbiological methods that do not utilize or validate using any live cultures, even a molecular assay, are integral in ensuring an effective and complete DNA extraction. Using only nucleic acids to validate a microbiological method are not validating a method that reflects the natural state of the target organism. Internally, we take great care in all of our method validations, including our microbiological methods, because we recognize the implication of the data that we generate. So thank you for your time and attention today. Please feel free to contact us at CW Analytical if you have any questions about anything that we've discussed today. And welcome, Kevin.
Hello, this is Kevin Moore. I am a Technical Sales Specialist for Bio-Rad Laboratories. I'd like to start off today with a quick company overview of Bio-Rad. Bio-Rad is a leading global provider of sophisticated products for life science research and clinical diagnostic markets. Founded in 1952 in Berkeley, California by husband and wife team David and Alice Schwartz, the initial slogan was to make life in the lab easier for researchers and accelerate the discovery process. Bio-Rad has a rich history of developing, manufacturing, and marketing products that enable discovery with a diversified and complementary mix of products and technology. Bio-Rad has shown continuous innovation with more than 65 years of strong performance. Bio-Rad has offices and facilities around the world with more than 8,000 employees and 150,000 customers worldwide. With headquarters in Hercules, California, Bio-Rad has a presence in nearly every significant geographic market across the globe. The vast majority of Bio-Rad sales are made directly through their own technically trained sales force. By 2003, Bio-Rad had over $1 billion in sales, and in 2008, joined the New York Stock Exchange. This slide shows the total sales in each of the global strategic areas for 2015. Bio-Rad's IQ-Check assays are based on gene amplification and detection by real-time PCR. The kits contain ready-to-use primers and probes specific to the target organism and utilize amplification of a DNA sequence that is specific to the targeted parameter. This amplification is based on the physical properties of the DNA combined with an enzymatic reaction. Detection is in real time using fluorescent measurement. Now we'll take a look at some of the key features of the Bio-Rad IQ-Check assays and what sets Bio-Rad apart from some of our competitors in this market. The IQ-Check assays use double-stranded hybridization probes that were developed by Bio-Rad scientists. These sequences are specific to highly conserved regions of the target genome and provide high sensitivity, high specificity, and an extremely stable configuration. The probes consist of fluorophores and quenchers on separate complementary strands as seen in the illustration. When not bound to target DNA, energy is transferred from the fluorophore to the quencher, therefore no fluorescence will be detected in the target channel, thus indicating a negative result. Each reaction also has an internal amplification control. This ensures no interference from inhibitory compounds and confirms the reliability of any negative results. The kits also include lysis reagent for DNA extraction. The lysis reagent is a proprietary resin formulated to bind and remove inhibitory compounds. The lysis step uses a three-tiered approach of mechanical, chemical, and thermal to lyse the cells. Because the IQ-Check assays are a closed-tube PCR system, you eliminate any post-amplification manipulation of samples and greatly reduce the chances of cross-contamination. The IQ-Check protocol is a simple four-step process. The first step is to enrich the samples. This is necessary to grow pathogenic organisms that may be present to a detectable level. The second step is DNA extraction. For this, you collect enriched sample and lyse the cells. The third step is PCR setup and amplification. For this, you add extracted DNA to a PCR well and start the PCR run. The fourth and final step is to evaluate the automated analysis and interpretation and report results. Bio-Rad's IQ-Check kits are available for a large number of pathogenic organisms in both qualitative and quantitative solutions. Here you can see the kits that are currently being used in the cannabis testing market. These kits include the E. coli 0157H7 assay, the Enterobacteriaceae kit, the Salmonella 2 assay. We have a couple of different kits available for Shiga toxin-producing E. coli, the Virex kit and the Sero kit. We also will soon be launching the IQ-Check Aspergillus assay. A typical cannabis lab setup to run Bio-Rad assays consists of the following things: the instrument and software, a thermoshaker, PCR kits, micropipetters, PCR plastics and consumables, and then a couple of other general lab items. Now let's take a look at the instruments used to run Bio-Rad's IQ-Check assays. Bio-Rad uses open real-time PCR detection systems. These can be used to run any type of PCR assay from virtually any vendor. The CFX96 Deep Well system is a 6-color, 96-well real-time PCR instrument that utilizes LEDs for both excitation and detection. The IQ-Check assays are also run using CFX Manager IDE software. This software includes templates with automatic call of results for all Bio-Rad IQ-Check assays. It can also be used to set up and run just about any other type of PCR experiment. There's also an optional automation system with Bio-Rad's IQ-Check Prep that utilizes Hamilton robotics. This system will basically set up and run all of your PCR assays. The highlights of the Bio-Rad system include the system is easy to install. It's a flexible solution that can be configured from low to high throughput depending on your sample volume. It's extremely user-friendly with only one window to set up and run PCR. And all IQ-Check food and cannabis tests can be run on the same plate at the same time. Here are some of the key features of the CFX Manager IDE software. You have automatic data analysis and interpretation of results, full LIMS capability to import and export data. There's an optional barcode reader for easier plate setup. Automatic email notification of results is also possible. You have improved data report for better traceability. There's an option for reagent lot tracking. And there are various security levels from admin with full rights to user with limited rights that can be incorporated into the software. Here's an example of a typical PCR run using CFX Manager IDE software in the IQ-Check assays. You have 3 different views of the data in the data file. The first is a data plot and graph to quickly view curves. The second is a PCR plate view file with a color change depending on the sample results. And then lastly, you have a detailed numerical data for each well with the CQ values for both target and internal amplification channels, and again, a color change depending on the sample result. In addition to the PCR data file from the previous slide, there's also a PDF report file that can be automatically generated for each run. The report file will contain all of the same information found in the data file, plus some PCR run details. Section 1 has things such as instrument serial number, the date the run was complete, QC lot information for the assays. Section 2 will give you a PCR plate view of your results. Section 3 gives you QC data for the negative and positive controls. Section 4 gives you, again, a summary of your results with CQ values for both the internal amplification control and the target channel. And then Section 5 gives you an overview of the curves and what they look like. Here we have the specifications for the Salmonella and S-Tech IQ check assays. You can see the specificity and sensitivity data as determined by numerous intra-laboratory studies have given very good results. Because of the extremely stable structure of the probes, kits are shipped at ambient temperature and only require storage at 2 to 8 degrees upon receipt. So no need to store any of the reagents frozen. They typically have a 2-year shelf life from manufacture date. They've been tested on a number of different cannabis matrices and shown to give very good results as well. You can see the number of different species that were used for inclusivity and exclusivity testing. And then these kits both have AOAC validation for use in the food industry as well. Bio-Rad also offers a line of chromogenic media that may be used for detection, confirmation, and enumeration for various organisms. Chromogenic media provides a quicker turnaround time than traditional culture methods in an easy-to-read format. Here are some of the medias that are available for use in the cannabis industry. Chromogenic media differentiates bacteria using chromogenic substrates and can be used for several different applications. Some reference methods call for chromogenic media to be used. They can also be used for enumeration of organisms. They can be used for differentiation, confirmation, and alternative detection methods. Some of the user benefits of using chromogenic media are that they are specific for the organism of interest, they're sensitive, easy to read, fast, affordable, and result in an isolated colony that may be used for further confirmation. The next topic we would like to present is the definition and significance of method validation. Method validation is the process of evaluating the performance of a method and demonstrating that it meets a particular requirement. There are several different reasons to perform method validation. The first reason is ethical reasons. A manufacturer wants to provide assurance to its customers on the accuracy of the results that they obtain by using their product. It's also just good science. There's also commercial reasons for product liability. A manufacturer should want to ensure that they are producing a product that performs as it is intended. And finally, regulatory reasons. To meet regulatory requirements such as those set forth by organizations such as AOAC and AFNOR. Validation studies performed on IQ check assays and rapid chromogenic media are conducted at independent laboratories to determine the performance characteristics of the method in comparison to a reference method, such as the USDA MLG, the FDA BAM, ISO methods, or Health Canada methods. Validations include inoculation with live organism, not spike DNA, as the method validation is not complete unless both extraction and detection for the assay are tested. The studies include probability of detection analysis, inclusivity, exclusivity, lot-to-lot stability, and robustness studies. We hope that you'll join us for our next webinar in August, where we will discuss microbiology in the cannabis industry. Is your cannabis testing method truly validated? Our featured speaker for this event will be Mr. Patrick Bird. Pat is an AOAC technical consultant and owner of PMB Biotech Consulting. That concludes the information that we wanted to present today. We want to thank everybody for joining, and we will now open the floor for any questions. Thanks for joining, everybody. If you would take a minute and complete the survey box on the left side of the screen. We've had a couple of questions come in, so we'll go ahead and answer those. So the first question is, do you need all 3 of the STEC genes to have a positive? My test only tests for STX1 and 2. So in the U.S., to be considered an STEC positive organism, you actually have to have the presence of the STX1 or 2 gene and also the EAE gene. So the STX genes are what the gene that codes for the Shiga toxin production, and then you also have the EAE gene, which codes for the attachment. So the bacteria has to attach to your gut, produce the toxin to make you sick. So yes, in the U.S., you do have to have the presence of Next one. Next question is, will we be able to run the Aspergillus test on the same plate as Salmonella and STEC? And the answer to that is yes, you will. Yes, they're going to be running the same thermal protocol, so they'll be able to run at the same time on the same plate. But the next question is, and this is one for CW, so the question is, why is enrichment necessary for pathogen detection?
All right. Hi, guys. This is Emily. So enrichment is necessary for pathogen detection, essentially to prevent things like false positives. Organisms like Salmonella can be easily outcompeted by other organisms, especially if the cells are injured. So it's to really ensure that there are no false negatives in your assay.
Very good, and we'll wait for the next question. The next question is, how does your internal control work, and is it present in every reaction? So the internal control is present in every reaction for the Bio-Rad assays. The way that it works is actually a proprietary internal control, so I'm not going to be able to discuss that. Just know that it is present in every well, and so basically you have to have an internal control to know that you have a valid PCR reaction happening in each well. So if you didn't have an internal amplification control, and you had a negative result, you would have no way of knowing whether or not PCR actually happened in that well. The next question is, are there any plans to implement a viability assay into the Aspergillus kit? So Bio-Rad currently has a solution for eliminating free DNA or dead cells. It's used in many of our other pathogen detection assays. So it's an application that you apply on the front end of your extraction, and it basically eliminates any dead cells that are there. So you should only be detecting viable cells that are enriched and are currently growing. And the next question. Next question is, how do you measure endofungal bacteria that live inside fungi with culture-based measurement? And I will let the ladies from CW tackle this question.
All right. I think that this is not an extremely straightforward question to answer because it would depend on the target, the endofungal bacteria specifically that you're looking for, to design your assay for the proper enrichment protocols as well as the proper target that you're looking for. So that, I don't know exactly how you would measure that, but it would have to be designed specifically around what kind of endofungal bacteria that you're looking for.
And so the next question, thanks Emily, the next question is, how much product is needed for PCR enrichment? So in most states, the requirement is 1 gram, at a minimum of 1 gram. So you would enrich 1 gram of sample, and then you use that for your extraction and PCR detection. The next question is your internal control then not a control for the extraction itself? How do you know your extraction was successful? Well, the methods are validated, and so the extraction is very it doesn't have a lot of steps involved in it. So if you have any kind of component that's inhibitory in the process, you're going to be able to tell whether or not the reaction happens. So, but because of the way the extraction is set up, there's not really much that can go wrong. And so these methods have multiple validations and have been tested by a large different number of samples. And so there's not really a good answer to that question other than the extraction itself is so crude that there's not really any way for it to not work. So the next question is, does the CO2 extraction clean the product from mold and fungus? And I will let CW answer this question.
Sure. So, you know, we haven't done any scientific studies to prove that that's what's happening, but that is what we suspect is happening due to the pressure in the extraction process. You know, it's suspected that it would kill any bacterial or fungal organisms that are present or inactivate any spores, but it's not something that's been, you know, there's no published research on that.
Next question is, are there current limits of detection for yeast and mold to give clients a pass-fail grade? So, CW, I'll let you answer that.
Sure. In the state of California, there are no limits of detection or action limits for yeast and mold to give clients a pass or fail grade. Before 2018, people would voluntarily choose to have yeast and mold limits on their product because it's a good quality indicator survey that can be done on products. And typically the action limit that was adopted was 10,000 CFUs per gram.
The next question is, what incubation time is best for fungi versus bacteria, or are they the same? So for Bio-Rad has an aspergillus assay that we will be launching here shortly. We'll have more details about that. As far as the bacteria goes, they're typically the same. It's usually going to be 24 hours. Aspergillus, the goal would be 24-hour enrichment as well. But until that kit is launched, until it's formally launched, and until that time, we won't be able to disclose that. Next question is, how long does it take to get results for PCR, enrichment, hands-on time, thermocycler? So you're looking at a 24-hour turnaround. So typically enrichment is about 20 hours, and then you have processing time, depending on the number of samples, take you an hour, hour and a half, and then you have a 2-hour run on the PCR instrument. So less than about 24 hours, maybe a little bit less than 24 hours. When will the aspergillus kit be available, ballpark? We're looking at end of July, early August, I believe. So that's the ballpark. See what the next question is. Our current method uses the cannabis DNA as the positive control. Is your internal control something other than something along these lines? Yes, it is. It's not using anything, it's not matrix related. So you don't have to, because these kits are used for various matrices, it's not dependent on what's actually in the sample. It's just depends on whether you have a valid reaction in that well or not. Are there any Pseudomonas chromometers? So this question is asking whether or not there's any chromogenic media for Pseudomonas. Yes, there are several different brands. Bio-Rad has chromogenic media for Pseudomonas that can be used if needed. And the next question is, what is your opinion on including Listeria in the testing panel? I'm not really, I think I'll let CW answer this question. One thing that I will say is that for any type of food manufacturing, food processing, those plants, those manufacturers are testing for Listeria in the environment. So I'll defer that also to CW and see what their opinion is on that.
Sure, Kevin, I would definitely agree with that. Any food product should have Listeria. We should really be considering the matrix type whenever we're designing our action limits and target organisms for cannabis matrices. It has not been investigated whether Listeria is a quality assurance risk on cannabis, but it's definitely not going to hurt to look for that.
Yeah, the next question is, are some of the other manufacturers in cannabis testing using nucleic acid only for their validation? They said they validated it. I'm not really in a position to discuss anybody else's validation. In August, we're having another webinar to discuss method validation. So I would suggest you tune in then and see what some of the experts have to say about validation. Next question is, do you ever see early amplification in your PCR run? If so, have you found out what caused it? Early amplification. So typically early amplification, I mean, that's not really, there's not really a good answer to that question. I would say for the Bio-Rad assays, you don't usually see early amplification in your PCR run. If you do see that, it's probably from a spiked sample of some sort. So, there are limits set up in the assay so that if you have a CQ value less than a certain number, so if your curve crosses a threshold before a certain number of cycles, it's going to be called invalid because it's just not possible for the DNA to amplify to that extent. There's not really a good answer to that question. I would say for Bio-Rad, we don't, you might see it on occasion, but it's typically due to a spiked sample or some kind of error during setup. The next question is, sorry I arrived late. Are the slides available? I believe they will be available later. I think this whole presentation will be available later, so it'll be on demand, so you'll be able to log in and see it then. And I think that's all the questions we have for today. So I'd like to thank everybody for joining and thank the ladies from CW for presenting. And hope that you'll join us in August for our next webinar. And have a good day. Thanks.
Thank you.